Mike received a bachelor degree in biochemistry at Brandeis University in 1998. After completing a PhD in RNA biology at MIT with David Bartel, he also finished a postdoctoral fellowship in biochemistry in David Liu's lab at Harvard University. And then he joined the Cancer Genome Analysis Group at the Broad Institute, led by Gad Getz, from 2008, first as a computational biologist and then as a group leader in computational biology, before he started his own laboratory at MGH in 2016, where he has been an assistant professor since. Mike has done really many of the foundational bioinformatic development and method development, algorithmic development that are still widely used in cancer genome analysis. One of the hallmark method that Mike invented was the MutSig, or mutation significance, which was used to identify recurrently mutated cancer drivers in all of the TCGA. Mike is extremely prolific. He's published more than 100 papers with a total citations of more than 200,000. I think that partially explains why when I mentioned about Mike coming here, a senior colleague of mine mentioned to me, is he a full professor yet? In addition to publishing cancer genome analysis papers, Mike has, several of Mike's recent works have really shown how elegant analysis of cancer genomes can turn into new biological insights that can then lead to further research questions in basic cancer biology. In 2016, Mike published a paper on Cell describing the two forms of mutation strand asymmetry related to transcription and DNA replication, where he actually first reported strand coordination of mutations generated by deamination by the family of enzymes known as the APOBEC. And he has since then built up a really interesting research program focusing on APOBEC mutagenesis, elaborating both the biochemical mechanisms of deamination by APOBECs, as well as its widespread roles in cancer evolution, including therapy resistance. So I think that's the topic of his presentation today. Without further ado, thank you, Mike.
Thank you, CZ. That was such a kind introduction. I've never had such a nice introduction before. And thanks for the invitation to speak today. I've really been looking forward to coming here today. So yeah, today I'm going to talk about APOBEC, which is a family of enzymes in all human cells, and how their activity drives molecular diversity and actually can directly drive cancer drug resistance in targeted therapy. So to start out, what is APOBEC? So it's a family of enzymes that are all cytosine deaminases. So that means that they attack the C base and take an amino group off it, replacing it with a keto group. So you have a uracil instead of a cytosine. And it can do this on DNA or RNA, but it always operates on single-stranded nucleic acids, and that's going to become important later in the talk. There's a pretty wide range of these enzymes in humans, and the APOBEC3 cluster is a group of 7 enzymes all next to each other on chromosome 22. Some are monomeric and some are dimeric, two cytosine deaminase domains in tandem in the same molecule. And this is actually a recent diversification in primates. Mice and rats only have one APOBEC3 enzyme, but primates have these 7 here. So something has recently changed here, and it's an active site of evolution. You can look through vertebrates and kind of see where, who has what APOBEC enzymes, and there's a correlation to what kinds of viruses each of these species have. Species with exogenous lentiviruses tend to have more copies of APOBEC. So these are essentially antiviral enzymes. They're part of the innate immune system. And their normal role in healthy cells is to attack nucleic acids that don't belong in the cell. But healthy cells tend to keep these enzymes shut down because mutagenizing your nucleic acids is a dangerous game. And, and it's something that cells don't want to do unless they're forced to. So the way APOBEC works, when there's single-stranded DNA or RNA, APOBEC can go in there and deaminate the cytosine base to give a uracil. And if the cell doesn't repair that properly, then you can get a mutation, a C to T mutation, or a C to A or C to G mutation. You can get changes to all three of the other bases. And this is just a molecular model. It's the yellow is the crystal structure of one APOBEC enzyme, one of the monomeric ones, APOBEC 3A. This is the most potent of the APOBEC family. And you can see there's this yellow base is the cytosine that's going to be deaminated. And in this picture, it's being shown at the end of this hairpin. This is a DNA hairpin, which can form whenever DNA is single-stranded in the cell. And of course, that has to happen when DNA is replicated and when DNA is transcribed. Basically, anytime something happens to DNA, the strands separate, and if there's a palindrome, it can fold back on itself and make a hairpin like this. Whoops. Let's see if we can get back to this. Okay. So as I mentioned, if these uracils in the DNA are not properly detected and excised and resynthesized as cytosine, then you get mutations. And actually, this is what got me into APOBEC as a topic. During the TCGA days, when people started sequencing thousands of tumors and looking at what kinds of mutations are there in each one, the APOBEC mutation signature really jumped out very clearly. Probably many of you have seen this plot. We call this the Gaddygram. This is all the tumors that we had sequenced as part of TCGA and other projects. Every dot on this plot is one person's tumor, and the tumor types are organized from those with the fewest mutations on the left to those with the most mutations on the right. So melanoma, lung cancer, these are tumor types that have the highest mutation rates, and these are associated with known mutagens like UV radiation in sunlight or chemicals in cigarette smoke. At the other extreme, you have childhood cancers and leukemias that have very, very few mutations. So the y-axis here, this log scale shows the total tumor mutation burden as the number of somatic mutations per megabase. And it ranges over 3 or 4 orders of magnitude. And the tumor types are sorted by their median tumor mutation burden. But you can see that within any tumor type, there's quite a range, right? Each tumor type kind of spans a large range there. So there's a lot of heterogeneity in total tumor mutation burden, but even more interestingly, there's heterogeneity in the kinds of mutations. And at the bottom of the plot here, these six colors show the 6 different ways you can mutate a base pair. If you use the convention that you describe what happens to the pyrimidine side of the base pair, then you can change a C to any one of the other 3 bases, or you can change a T to any one of the other 3 bases. So you have these 6 kinds of ways, 6 kinds of mutations. And you can see that in melanoma, it's almost all this yellow color, C to T mutations, whereas in lung cancer, you've got a lot of this cyan color, which is C to A mutations. We make these Lego plots that you may have seen before. This is kind of a two-dimensional version of the one-dimensional plot that you sometimes see in papers, but having it in these two dimensions like this lets you see patterns where either the 3' base is distinctive or the 5' base is distinctive. This plot that I'm showing here, this is the most common kind of mutation. It's the most common kind of mutation both in cancer, in somatic mutations, as well as in germline mutations in evolution. And that is C to T mutations when the C is followed by a G. And the reason why this is such a mutable context is because the CG dinucleotide, whenever you have the sequence CG in vertebrate genomes, it's methylated on the C, on both strands. And so having that methylated C makes you one step closer to a T, a thymidine. And if you deaminate methyl C, you actually have T now. And it's not if you deaminate regular C, you get uracil, and uracil sticks out as not belonging in the DNA. But if you deaminate methyl C, you get T, and that does belong. So it's harder for the cell to recognize it as a problem. So that's why CG dinucleotides are so mutable. And they're going extinct in vertebrate genomes. There's only about 10% of as many as you would expect from a random distribution, because they're gradually dying away in evolution. So this is the most common mutation type in cancer, and it's in the Sanger Institute's catalog of mutation signatures. It's called signature 1, SBS1, single base substitution 1. The second most common kind of mutation in cancer is APOBEC mutation, and signature 2 is this APOBEC signature. Signatures 2 and 13, they're called for kind of historic reasons. But you can see that there's this back row pattern here, where now you have C's being mutated when they have a T before them. And that's the hallmark APOBEC motif, TC. Wherever you have TC in the genome, that C can be a target for APOBEC, in particular the APOBEC3 enzymes, APOBEC3A and B. So this back row pattern on the Lego plots is what we've learned to recognize as the APOBEC signature. And as I mentioned, the C can change to any one of the other 3 bases. The C to T mutations overlap with a lot of other signatures, but the C to G mutations don't overlap so much with other signatures. And whenever you see these 2 red fingers here, that's really the classic sign of APOBEC. And so we can go back to the gaddygram, and now instead of just sorting by, from leukemias to melanomas, we can cluster the tumors so that they form these natural groups dominated by particular mutation types. So like, for example, here you see melanomas in black that have this C to T signature. There's actually some tumors that are even more highly mutated than melanoma, and that's when you have mutations in the polymerase epsilon that lose the ability to proofread. These are called pol mutant tumors. There's stomach, endometrial, those are some of the tumor types that have that. And then you've got the lung cancers here that have this G to T or C to A smoking signature. But you can see that here there's this multicolored group of tumors that have the APOBEC back row signature. And these tumor types are cervical, bladder, head and neck, breast, lung, and to a certain extent, smaller numbers of other tumor types. But these are really the big 5 for APOBEC. you can sort of order the tumor types by how prevalent APOBEC is in them. For cervical, it's pretty much every tumor is dominated by APOBEC. This plot down here doesn't really match that, but almost all cervical tumors are HPV positive, and that seems to be related. Okay, so TCGA was all primary tumors before any treatment, and looking kind of broadly across all tumor types, we see that about 50% of primary tumors have APOBEC mutation signature to some extent. So this is just kind of toy data here. You could imagine, half of patients, their primary cancer will show some amount of APOBEC. What we've learned in more recent years is that when you focus on resistant cancer, so the tumor that comes back after treatment, that percentage goes up. And you have these examples of patients that had no APOBEC in their primary tumor, but when their resistant cancer came back, suddenly there's APOBEC. So this is intriguing. And I'm going to go into an example, an MGH patient, where we see very clear evidence that APOBEC is helping the cancer to find drug resistance mutations. So this was a paper that was published in 2023, led by Aaron Hada's postdoc Hideko Isozaki. The patient that was really the central illustration in this paper was this MGH086, a lung cancer patient that the initial biopsy found an ALK fusion. And so they got the first generation ALK inhibitor. But the whole exome sequencing of that first biopsy showed really no sign of that back row of mutations, the APOBEC signature, and also a kind of a low number of total mutations, about 60 somatic mutations in the exome. So because the patient had a tyrosine kinase driver, they were put on crizotinib, the first generation TKI, and the therapy was successful. The tumor went away for about a year, but it came back. And this is the all too familiar story with targeted therapies. The cancer finds a way around the therapy. So we did a repeat biopsy and compared what it looked like now. And what we saw was there were almost 800 new coding mutations on top of what was there in the initial biopsy. And as pure an APOBEC signature as you can imagine, just this back row of mutations. And one of those mutations was the well-known ALK resistance mutation, ALK E1210K, that's been shown in very careful lab work to confer resistance to crizotinib. By that time, brigatinib was available, second generation tyrosine kinase inhibitor. So the patient was put on brigatinib and was cancer-free for a few years, but then the cancer came back actually in 3 different anatomical locations, 3 independent resistant tumors. And each one of those tumors was biopsied and got whole exome sequencing. And amazingly, what we saw was that there were 3 different solutions that the cells came up with, 3 different double mutations in ALK. Each one of them added a different on-target mutation in ALK to become resistant to brigatinib. And this was all in the context of continued APOBEC mutagenesis. All the new mutations are APOBEC signature, including these ALK driver mutations. They're all at this TC motif. So after a few more years, the patient got put on lorlatinib, which became available, third generation tyrosine kinase inhibitor. And that worked for a while, but after less than a year, the tumor came back, now with a triple ALK mutation in the context of almost 600 new mutations, again, with this APOBEC, pure APOBEC mutation signature. So it's really, it's really tempting to interpret this as the cells had no need for APOBEC in the beginning. They had all the driver mutations they needed, but once the cells were put under pressure with this targeted therapy, then they used this tactic of turning on APOBEC, getting this random hypermutation throughout the genome, and just letting this selection process happen. Any cell that happens to get a resistance mutation is going to grow out and seed the resistant tumor. So we really want to we want to stop this from happening, because these targeted therapies represent many years of many people's work, and it's discouraging to see cancer get around it so easily. Okay, so this is just kind of another way of showing the same data for this MGH086, this branching evolution where the branches are colored by how much APOBEC mutation there is among the new mutations. And it's not just this one patient. Here's another lung cancer patient, also an ALK fusion-driven cancer. All these early biopsies didn't have any sign of APOBEC, but once the patient was put on lorlatinib, that's this last branch, they suddenly turned on their APOBEC and just got all these new mutations, including ALK 1196M. That's a known lorlatinib resistance mechanism. This is an NTRK-driven cancer, same pattern, APOBEC coming on in the late branches of the evolution. So this is a pattern that we see. If you think of it as the cell kind of, randomizing, shuffling the deck, it's looking for a lucky mutation that's going to confer resistance. And so this is what we would like to have some way of impeding. And so kind of until now, there had been a question in the field, like, we see all these APOBEC mutations, but are they really causative? Are they really doing something, or are they kind of an epiphenomenon? Are they just something that's also happening in the cell during the cell's journey to a resistant state? We really wanted to kind of lock that down, and this is where Hideko's work in Aaron Hada's lab really, I think, for the first time sealed sealed the case for APOBEC being causative in resistance. So Hideko uses this cell line model, PC9 cells, which are a lung cancer cell line driven by an EGFR exon deletion. These cells, divide like crazy on the dish, but if you treat them with a TKI like osimertinib, most of the cells die. And what Aaron actually worked out during his early career was that if you wait long enough, you can get them to come back just by waiting long enough with this particular model. If you wait 6 weeks, you'll get drug-tolerant colonies emerging on the plate. And then if you look at those to ask what's different about them molecularly, one of the prominent gene expression changes is APOBEC3A getting upregulated. Actually pretty quickly, like within the first day, you can see APOBEC3A turning on. And that's not true for the other APOBECs. So this was an important piece of evidence, kind of 5 to 10 years ago, as the field was really trying to work out, is it APOBEC3B as was initially claimed? It turns out APOBEC3A is probably the one doing it. So we see this in these 3 different models of TKI inducing APOBEC3A expression. So we can see the messenger RNA going up. We can also see other effects in RNA. So I mentioned APOBECs can act on RNA as well as DNA, and this is a form of RNA editing. So there's two main kinds of RNA editing, and probably the more famous is A to I editing, adenosine to inosine editing. This is RNA editing catalyzed by the ADAR enzymes. But there's also C to U editing, and this is done by APOBECs as well as AID, which is a very closely related enzyme that works in the adaptive immune system to diversify antibodies. So APOBECs can do this RNA editing, and in collaboration with Li Zhou and Remy Buisson, when he was in Li's lab, we found one particular hotspot in the gene DDoS, DDoST, where the messenger RNA folds up into this hairpin, and then this C is a super hotspot for RNA editing. And we can monitor APOBEC activity, actually the momentary APOBEC activity at a given moment by looking for this RNA editing. The DNA mutations are kind of a historical record of the APOBEC activity that happened in a cell, but the RNA is always turning over, and so you only see this RNA editing if there's active APOBEC in the cell, kind of right then, right when you kill the cells. So do we see that in Hideko's model of these emergence of these drug-tolerant colonies? They're expressing APOBEC. We also see that they're doing this RNA editing. We can quantify the percentage of messages that have this DDoSed point change. And actually, Remy developed a digital droplet PCR assay that measures this very sensitively. So this is very useful. And you can see that by 3 days of gefitinib treatment, you've got 20% editing at this DDoS hotspot, and we see it going up in all three of these models. So APOBEC 3A starts getting expressed. You start getting this characteristic editing. These are 3 more models with lorlatinib as the treatment. We see this increase in editing there too. And also this is RAS. This is an anti-RAS drug, an old one, trametinib, and this new one that targets the G12C mutation. We see it here too. So these multiple targeted therapies are all inducing APOBEC 3A, not APOBEC 3B, and causing this RNA editing to become prominent. So definitely, this is something that's happening as a consequence of therapy. It's not just lung cancer. In a collaboration with Dan Faden at Mass Eye and Ear, we've been looking at head and neck cancer, and a few years ago we published this story about one patient where we had 3 different biopsies from the same patient. It's single-cell RNA, and so we're trying to follow the evolution of this person's head and neck cancer kind of longitudinally. And there were changes in gene expression, but there was also the emergence of RNA editing. There was no sign of this characteristic DDoSed APOBEC event in the initial biopsy or the initial lymph node biopsy, but the secondary tumor that formed did have this RNA editing. So again, we see a weak or no APOBEC activity in the early stages, and then APOBEC turning on in the late stages. Okay, so this is just kind of a schematic of the point that I was making, that the APOBEC mutations in the genome are kind of an integration of all the APOBEC activity over the cell's natural history. But when you see RNA editing, that means there's active APOBEC right now. So we see DNA mutations, we see RNA editing, how do we know that it's actually doing something? So this is where Hideko's work really was important. So here's an experiment. This is a different cell model. This isn't PC9. This is a patient-derived cell line, MGH119. You treat it with gefitinib, all the cells die, and in this model, you can't just wait for colonies to emerge. You wait and wait and wait, and the plate just stays empty. The cell line model has a harder time evolving resistant colonies. And it's an interesting question, why is that? So Hideko introduced APOBEC 3A into the cell line, trying to ask, can we accelerate the evolution of this cell line? And when she treats the cells with DOX to induce overexpression of APOBEC 3A, indeed, she sees resistant colonies emerging during the same amount of time. So you add APOBEC and you can get resistance. And crucially, the control experiment she did was expressing this point mutant of APOBEC 3A, E72A, which has no catalytic activity. So this is a dead version of APOBEC. You express that, and you don't get this effect. You need catalytically active APOBEC 3A. She also saw an increase in the amount of resistant colonies with that PC9 osimertinib model. So this is looking at lorlatinib treatment of this H3122 cell line. This wasn't a kind of a nothing or something difference. It's more of a subtle acceleration of resistance when you overexpress APOBEC, but you don't see the effect with the catalytically dead mutant. So that's the phenotype. You get more resistance. This is the genomics of it. Did I skip this? No. Okay. This is where Hideko's doing the opposite thing. She's CRISPRing out the native APOBEC 3A in PC9. And so she's doing a APOBEC 3 knockout versus a scramble control. And what she saw is that in the scramble control, when you take the resistant colonies that emerge and do whole genome sequencing on them, you see all these mutations. Many mutations, they tend to be in clusters or contiguous, and you also see structural variants. That's what these are here. But when you directly compare the CRISPRed version, suddenly the cells that survive the treatment, they don't have this increased mutation burden, they don't have clustered mutations, and they don't get as many of these structural variants. So it's kind of all the pieces of the story. We're seeing the genes, APOBEC's being expressed, it's acting on RNA, it's acting on DNA, and it's actually causing the cells to become resistant faster. So this is in the CRISPR experiment, in the PC9 osimertinib model and the 3122 lorlatinib model, with the scramble condition, both of these models can evolve resistant colonies, but when you knock out APOBEC 3A, it knocks down this ability quite substantially, and the cells really are much slower at evolving resistance. And then when Hideko took this knocked out cell line and added APOBEC 3A back with overexpression, she rescued the effect. So really, in both directions, kind of the positive experiment and the negative experiment, she's showing that APOBEC 3A is causative in the resistance of these models. So this really makes us eager to find some way of inhibiting APOBEC. And there's questions about how good of an idea would that be? I already told you that APOBEC is antiviral, so So if you treat someone systemically and knock down their APOBEC ability, you're really ablating part of their innate immune system. And is that going to make them more susceptible to viruses? And what's the trade-off? Virus versus cancer. These are important questions that people are asking. But kind of with this this casino metaphor that's due to Daniel Haber, we really are looking to have an advantage against the house that's really dealing all these random hands. And we'd love to be able to, at the same time as giving targeted therapy, we'd love to be able to give an APOBEC inhibitor so that, at the same time that the cells are challenged with this precision medicine, we're also shutting down one of the tactics they can use to quickly evolve around it. We don't have APOBEC inhibitors yet, and this is an active field that people really are working hard in. There's been small molecule library screening done without much success, but the idea here is that if we in our model system here, we've shown that if you knock out APOBEC, it knocks down this ability for these drug-tolerant persisters to emerge on the plate. What if we could do that with an inhibitor instead? So that's the idea, and the hope is that this could delay the emergence of resistance, make the patient survive longer, and really prolong the benefit of these precious new medicines. So what could be a rational route to an APOBEC inhibitor rather than the usual approach of just screen thousands or millions of drug-like small molecules? This is where it gets kind of interesting because we've learned a lot about the substrate preferences of APOBEC enzymes, and we can have some clues there about how we might even be able to design an APOBEC inhibitor. So this is where I'm going to go back to the tumor data. What I'm showing here is a list of the most common somatic mutation hotspots in cancers that have APOBEC, the APOBEC mutation signature. And there's a lot of recognizable names on this list. All these ones in gray here are kind of well-known cancer drivers like PIK3CA, TP53, ERBB2, et cetera. But there's a few in this list that are not famous driver genes, like this TBC1D12 and this MB21D2. And when we were first looking at this, it was one of those situations where either we found some new drivers, or these are artifacts, or something else. But one thing we noticed, we were thinking about hairpins already because of what was known from RNA. And actually, I remember when my MGH colleague Cyril Bennis came and talked to me, and he said, "Is it like that in DNA too?" And the idea really came from him. And when we took a look at these sort of non-driver hotspots, we saw right away that these are all potential hairpins. So you can take a short DNA sequence, put it in M-fold, and ask it to find the best secondary structure. May not be true, but it's kind of a starting point. For a typical driver like this PIK3CA hotspot, when you put this into M-fold, it's going to find some kind of structure. But what we typically see is that the mutation itself is just in an ordinary region. It doesn't tend to be in a particular kind of structure. It's usually in one of these single-stranded kind of situations. But here's what this MB21D2 hotspot looks like. It's at the end of this perfect hairpin, this 9-base palindrome with a 3-base linker, and the mutated C is at the 3' end of the linker. I think people who know a lot about nucleic acid structural biology say that the 3 nucleotides is the shortest loop you can have. That's the tightest turn that the backbone can do. So this is kind of the shortest kind of loop in a hairpin. One of the interesting bits of data here was that this mutation can either be a missense, a Q to E, or a nonsense, a Q to stop. And they're about equally balanced throughout TCGA. There's 5 bladder patients, 11 lung patients. There's all these different tumor types have this hotspot, and half of them are missense, half of them are nonsense. And that was always a puzzle when we were trying to think, how is this a driver? And what we saw was that there's basically this dichotomy. Known driver mutations, they're just at ordinary places in the genome, not any particular kind of secondary structure. But there's this other class of APOBEC hotspots that are all these hairpin loops. The third position of a 3-base loop, and the longer the stem, the better. And we think that this is because it kind of prepays the entropic cost of bending the DNA backbone there into the shape that the APOBEC enzyme likes. It's got, active site pocket, and to fit a substrate in there, you have to kind of bend the 2 ends back, and in a stem loop, that's already done. And so all it has to do is just bump into the active site, and it's like candy for APOBEC. These hotspots are so prominent, but we think they're passenger hotspots, and this actually is what convinced us that passenger hotspots is really a thing. It was kind of thought of as a contradiction in terms in the early days. It was thought that if you see the same base mutated over and over again across patients, that must reflect selection. What we think is these are just easy substrates for APOBEC, and they may actually have no causative role whatsoever in cancer. So I mentioned the C needs to be the last base in the loop if it's a 3 nucleotide loop. This is looking at relative mutation rate across many whole genome sequenced tumors. The x-axis is the hairpin strength, and we have the simple metric of just 3 times the number of GC base pairs plus 1 times the number of AT base pairs. This was a decent fit to the trend. And what we can see is that as the hairpin gets stronger, you see this really exponential increase in the mutation rate of Cs that are at the 3rd position of the loop, but no effect for the other 2 bases, if the C is in the middle or the other side. That's really evocative about what the geometry is there. It suggests that the hairpin needs to go into the active site in a particular rotation, right? And if you rotate it just a little bit away, the C is now not positioned correctly in the active site. This is an effect that's really unique to APOBEC. We don't see it anywhere else we look, any other kind of mutation signature. So the position is very important, but also the loop length is very important. This is the data for the 3 nucleotide loops. And as you go to longer loops, you can just look for these loops in the genome. You can find all the places that can form a 3 nucleotide loop or 4 nucleotide loop. The effect gets smaller as the loop gets longer. And for 4 nucleotide loops, the 3rd or the 4th position, these both show effects. For 5 nucleotide loops, it has to be the 4th position. So there are these interesting sort of subtle preferences. And this is just kind of showing the data in another way. The loop length is the successive layers of this pyramid, and then within each layer, it's the position of the mutated cytosine. So the red is where there's a strong effect. The mutation rate goes up exponentially with loop strength. So we see it for positions 3 of 3, 3 of 4, 4 of 4, and you can kind of see that the shape of the preference. And so after staring at this data for a while, we kind of wanted to come back to the idea of making an inhibitor. So we know from the crystal structure of APOBEC3A that the DNA substrate is bent into a U shape there. That was the only part of the DNA visible in this X-ray structure. But you can model a hairpin onto that just very easily. Take a hairpin crystal structure, it superimposes onto that shape there, crucially with the labile cytosine being in the right place, because it's we know from the structure that it flips out into the active site there. So we all know that DNA and RNA are not good drugs in themselves because of delivery issues, like how do you get them into the cell, how do you stop them from being degraded. But maybe we can sort of take some baby steps here and use this as kind of an inspiration for one mechanism for inhibiting APOBEC. So Ajinkya Kawale in Lee Zhou's lab, when Lee was still at MGH, he did this very simple proof of principle experiment where he took a model of APOBEC activity where you have the substrate being deaminated and then cleaved to give the product. That's when you add APOBEC3A containing cell extract. And then he used a DNA hairpin as an inhibitor. And it's really just a competitive inhibitor in this experiment, but it's the kind of the simplest proof of principle. As you add that inhibitor, you see the activity going down. And we could imagine making ever better substrates by for example, we could close covalently close the two ends to stop exonucleases from getting in and degrading it. We could introduce phosphorothioates to try to increase stability and resistance to endonucleases. There's also changes at the cytosine itself that we could think of. And there's one in particular, this paper came out a few years ago where they look at zebularine as a cytosine analog. So zebularine is cytosine without a leaving group at that position here, the position that is either an O in uracil or an NH2 in cytosine. It's just a hydrogen, so there's no leaving group. And when the enzyme binds to this kind of a substrate, it can add water here to make the tetrahedral intermediate, but there's nothing else that can get kicked out. So it's kind of a transition state mimic. And this is a result that Ajinkya got in his experiments using DNA oligos as an affinity reagent to pull down APOBEC3A out of a cell extract. When he does this with a hairpin that has uracil or cytosine in it, he's not able to enrich APOBEC3A from the cell extract. And that's also true with zebularine. If he just replaces the cytosine with zebularine, still no effect. But he was absolutely amazed that if he uses methyl zebularine, where this R group here is a methyl group, this paper that I mentioned here, they use a fluorozebularine here. We found it easy. Ajinkya found it easier to get his hands on methylzebularine instead of fluorozebularine. And if he just makes that one change, suddenly he can pull APOBEC3A out of the cell extract. So there's this modified oligo binds strong enough to the APOBEC 3A that you can fish it out of a cell. This is really encouraging, and Ajinki is doing follow-up experiments here, and eventually we want to apply this to Hideko's models of resistance. So that's where I'm going to end. This is kind of a work in progress, but I really like this story because it's an example of how you can go from kind of nerdy observations about mutations in cancer and mutation signatures to really concepts and ideas about how you could inhibit an enzyme and maybe pump the brakes a little bit on cancer's ability to evolve and outwit us. So I'm gonna end there, and I'm gonna acknowledge the people in my group who've done the work, as well as members of Esther and Doa's lab, who are always, we have our joint lab meetings, and we're always bouncing ideas back and forth, as well as the other labs that have been my collaborators and my support. And thank you all for your attention, and happy to take questions.